That's a good question, because sometimes I put things online and students think you don't have to come to class, because that happens too, but.
I just don't want to miss anything.
Yeah. If I ever do cancel class, I'll make that very clear. Class is canceled. But if I don't say class is canceled, you should clarify. We'll get started here. We'll talk about the integumentary system. Now the organs include glands, hair, nails, skin. Now skin is primarily what I'll be talking about. But there's other things that you kind of see as you study the integument system, which is mostly your skin. There's also associated things like blood vessels, nerves and adipose. Nerves especially because the skin is your largest sense organ for touch. And the major functions include, well, mostly protection. Skin is a physical membrane, a barrier. Let me advance the slide here. Get a picture of skin. I'll say against abrasion. Dehydration, because the keratin is waterproof. Think of it as a physical membrane barrier to pathogens. Remember, it's a cutaneous membrane. Also for sensation. We'll kind of look at different nerve endings that are in the dermal layer. And we also say thermoregulation. For example, it's not hot today, but on a hot day, you sweat, put moisture on skin, and it cools the body down. Okay, so for example, sweating can help regulate temperature, but also the fat in the sub-Q layer, or the hypodermis. I'll just say fat insulates. And also metabolic. Because again, the adipose or fat in the sub-Q layer can burn that for fuel. So you have fat. With exposure to sunlight, the skin can produce vitamin D, so it has some metabolic function too. So those are the basic organs and functions. And I think we should study the structure. They always show like the cube of skin. And this is a good time to mention the app, I was on the syllabus. If you go to models, for those of you who use it, there's a skin model under microanatomy. And this is kind of the same as the figure that I just showed you. A little darker. Okay. Wait, let's look at the organization of skin. So we just kind of draw a big cube. We can kind of see the superficial and deeper layers. I'll use green for adipose, kind of the bottom layer. But the top layer is the keratinizing stratified squamous epithelium. We just call it the epidermis. Top layer. I'll put epi for short. Epidermis, our stratified keratinizing stratified squamous epithelium. If I highlight it on the model, it gives you the superficial and deep layers of the epidermis, but it's both epidermis. Is that superficial layer of the epidermis there is the keratin. Kind of subdivide. Remember that the epidermis has a keratin. But the layer deep to it is called the dermis. And it's the thickest layer of the three. It's that whole middle layer that's highlighted in green. It's highlighted in green, but I'll use red to symbolize collagen fibers. The collagen fibers at the top of the dermis, which is pretty much from here to here. It's more of a loose CT at the top, and as you go deeper in the dermis, it becomes more of a dense, irregular connective tissue. So, more up here, it's a loose CT, and deeper on, it's more dense. But I won't draw too much, because there are other things I want to draw in here. But let's remember that the dermis is our example of a dense, irregular connective tissue. Okay? But at the very top, it's not dense, it's loose. That's what I'm saying. So, it's also dense, irregular. CT, connective tissue. And there are a lot of things in the dermis. For example, the model shows pretty well that there are blood vessels that start in the bottom layer. see if I can highlight one. So I highlighted in purple an artery that starts in the hypodermis, which is the fatty sub-Q layer, and it sends like a little branch to the top. So if you follow the blood vessel, I don't think highlighting it helps. The blood vessel goes all the way as high as it can, right underneath the epidermis. That's as far as it can go. Okay. Because remember, the epithelial tissue is cell packed, so there's no room to let the blood vessel in. So you have a blood vessel down here. It sends like a little branch all the way to the top. And then there's gas exchange, so that changes from red to blue. I'll just draw blood vessels. So you have blood vessels that are in the hypodermis. So pretty much from my bottom layer there, I'll just put hypo here, which is short for hypodermis. So I've shown you 3 layers for the integumentary system. The top layer is the epidermis. The bottom layer is the hypodermis, and the middle layer is the dermis. So those are all 3 layers. And when we say the term skin, we're referring to the top 2 layers, epidermis, as well as dermis. Okay, and know what tissue types are in each layer. The epidermis is our stratified squamous epithelial tissue. It's keratinizing. The hypodermis is mostly fat, and the dermis is our connective tissue, dense, regular, and loose CT. Okay, there's some other things here. So that's the basic organization, but let's draw the other things that we see. Here, you got a big old hair follicle. Blue hair, I drew a blue hair. Basically, the hair follicle, the living cells are in the dermis. I'll describe the anatomy a bit more carefully here, but that's a hair follicle. And there's a gland associated with the hair follicle we want you to know. It's called the sebaceous gland. I'll put a little star next to that. So the sebaceous gland is associated with the hair follicle. Its secretion is sebum, which gives hair that nice sheen, or kind of waterproofs the hair. Also associated with the hair follicle is a smooth muscle called erector pili. I'll draw a little muscle there. You can see it on the model too. It's this big muscle right there. Erector pili is the muscle that makes your hair stand on end when you get scared, like in a sympathetic response. So those are both associated with the hair follicle. The model shows it pretty good. Here's the sebaceous gland there. And there's sebum being secreted onto the shaft of the hair follicle, of the hair itself. We also see sweat glands. There's a sweat gland that's associated with the hair follicle. Let's see if I can find one here. Let's see. Yeah, that one doesn't highlight this gland here. It's a coiled tube. It's an apocrine sweat gland, and what I want you to see how the end of the gland, it terminates on the hair follicle. So this is a kind of sweat gland, apocrine. Secretes sweat onto the hair follicle. So it's using the hair follicle to get the sweat to where it needs to go. Sweat on the hair follicle. The sweat includes some fat and protein in it. PRO is my shorthand for protein, and you usually find these sweat glands in kind of anal axillary regions. Anal axillary regions. Armpit, you know. And because the because the sweat has fat and protein in it, the bacteria that live in your armpit kind of metabolize that, and we kind of know it as body odor, right? Other sweat glands don't have fat and protein in it. For example, let me show other sweat glands here. Here's one. This is an eccrine sweat gland. So the sweat is basically a hypotonic fluid. Basically, it's saline. There's no fat or protein in it, so it shouldn't generate that odor. These are pretty much everywhere else. Okay. Everywhere. That's a simple way to describe where they are. Just not the anal axillary regions. Sometimes they're called merocrine sweat glands. That's the term I use. Eccrine, merocrine, that's kind of the same thing. So let me draw a couple of glands on here. So let's remember that the apocrine, they're coiled tubes. Their duct will terminate right onto the hair follicle. So I'll put A for apocrine. But the merocrine sweat glands, they're coiled tubes. They'll go right to the surface. Okay. They don't have to empty on the hair follicle. So I'll put M for merocrine. In addition to the sweat glands, the nerve endings are usually in the dermal layer. You usually see them pictured here. I'm going to zoom in on this one. These are pretty good. It's tactile corpuscles. Let me see how much detail they show you. I don't know. I can't get that close. So what I've highlighted in purple are called tactile corpuscles. They're nerve endings that go right up to the epidermis. Okay. And Okay, let me draw it big on the board here. Okay, picture a nerve ending. Sometimes they're bare. Sometimes they have encapsulation. If it's a nerve ending that's encapsulated, what that really means is it's a little harder to stimulate that nerve because it has a covering around it. So if you want to physically poke it to stimulate it, you have to poke a little harder because it's got a capsule around it. However, because these go right up to the surface, right underneath the epidermis, they're not that hard to stimulate. So let me draw a little thing going right up there. These are called tactile corpuscles. And tactile corpuscles, they convey the sense of fine touch. It's kind of what the name of the sense. So you have the sense of touch, but there's all these subcategories. These nerve fibers are for fine touch, like if, you know, like you have them on your fingertips. You can feel the difference between sandpaper and silk if you're blindfolded, right? You can perceive that because of these types of tactile corpuscles. See, I want to look for one called that's deeper here. Oh, this one, that's what I'm looking for. That big one there, I highlighted the layers around the nerve ending. This is called a lamellated corpuscle. So what you see there is you have a nerve ending that has many layers around it, not just one layer like the one I just drew, but many layers. Lamellated corpuscle. And also look at the location. We're pretty deep in the dermis, right next to the hypodermis. So it's deep. And also it has many layers. So based on how I'm teaching this, is it harder or easier to stimulate that nerve ending? Harder, right? It's like deeper and there's more layers around it. So if you were to stimulate it, usually deep pressure and vibrations can trigger it. Okay? So that's kind of what it's for. To sense deep pressure, like from your masseuse vibrations. So let me put like a nerve ending way down here, put many layers around it. It's kind of where the lamellated corpuscles are. They show nerve endings that have no encapsulation. They're just called free nerve endings because that's exactly what they are. I highlighted some in purple there. So if you have nerve endings that have no encapsulation, that's what you just call them. Free nerve endings. They sense pain and temperature. I'll just draw a little nerve ending here. They're all in the dermis. That's why the integument is your largest sense organ because all these nerve endings are there for different sensations of touch. What I like about this model is it kind of shows how the epidermis is associated with the dermis. If you rip it apart like they did, it's not a smooth on smooth interaction, is it? The deeper layer of the dermis has these ridges. When I highlight it in purple, those little things are called epidermal ridges, and they interact with the bumps on the dermis, which are called dermal papillae. Okay? So that's how the two interact. So when you have this kind of ridge-papillae interaction, it's a snugger fit. Okay, it'll not tear so easily. So that's why I drew it all bump wavy like that. So what you call, on the epidermis end, call them epidermal ridges, and they interact with the things that stick up. It's hard for me to show it on here, that's why I showed it on the model. The dermal papillae. So I'm going to move away from the micro model. Is there any questions? Does anyone actually use the app? Okay, some people do. If you have any questions on, how to use it, I can help you, but it's pretty much self-explanatory. I think the students can handle it pretty well themselves. Well, I'm going to go back to this slide, and I'm going to just skip it now, right? Because I taught everything I wanted to using the app. But there is some slides that show us the organization of the epidermis that the app doesn't show. The different strata of the epidermis. California is like going to Santa Barbara as a memory jogger to memorize the different strata of the epidermis. So there's corneum, lucidum, granulosum, spinosum, basale. So call this strata of the epidermis. It's just a keratinizing, stratified, squamous ET. That's what kind of tissue it is. But we subcategorize all the different layers within this to kind of show you how eventually at the top you get these dead keratin-filled cells that are constantly shed. And I put letters, they put letters next to where the strata are. And I labeled this slide to kind of show from top to bottom of this epithelium how you can identify the different strata. But the picture shows it pretty good. Let me get my pointer. So now we see the epidermal ridges and the dermal papillae at the bottom, but the bottom layer of cells, and you literally call stratum basale. So I just point to the bottom layer, okay? But it's any bottom row is all considered that strata. The theme here is that from bottom to top, at the bottom you have living cells that are undergoing rapid mitosis. And what you're doing is you're trying to match the rate of mitosis to the rate of desquamation. It's shed at the top, and this rate of mitosis will eventually replace the cells you're losing. Okay? So as you kind of get pushed to the top, somewhere around here, cells start to die. And but the cells stay linked together, and it forms the stratum corneum at the top for waterproofing. So let's look at each strata individually. I took a close-up picture here so you can kind of see the nuance between the different strata. But let's start with the bottom one first to give you some notes on it. Now, stratum pistale, the base layer. Okay, but it's also called germatitivum. Germatitivum refers to the fact that it's a germ layer. This is where the young cells that we call keratinocytes. Keratinocytes. They undergo rapid mitosis. And like I said, I'll write it again, this rate of mitosis it matches the rate of desquamation. Shedding, the natural shedding of the skin. Now there's other things here. They show you melanocytes and tactile cells. So there are cells in this bottom layer. On the picture, they're right here. This bottom layer. There's a tactile cell. It's interacting with the nerve ending for more touch sensation. Okay, so you should note tactile cells. The tactile cells are not nerve cells themselves, is what I'm trying to say. But they stimulate nerve endings, basically. The other row, the other cell type in this row are the melanocytes. On the illustration is the gray cell. They have vesicles of melanin. Now, melanin give your skin pigmentation. And they help protect against UV. So I'm just writing protects against UV, gives skin pigmentation. The melanocytes, so they're in this bottom layer here, and the cell has these cytoplasmic extensions. So I'm going to just draw a cell with all of these extensions. The extensions are spreading the melanin to upper and lower layers. And so the higher up it goes, the darker your skin will be, okay? The darker your tan will be. Here's an illustration of the melanocyte. Much better than what I tried to draw on the board there, but they do show you the histological picture on the other side. And it's labeled M on the slide. And you should be able to identify that cell. Notice how it has a clear rim of cytoplasm around it. And that's usually how you spot them. And I looked on the picture that I took. I'll turn out the light so maybe you can see it a little better in the back. So be able to identify that cell in a picture like this. So that's the bottom layer. We'll move on to the layer right above it called stratum spinosum. Now, stratum spinosum is called the spiny layer, right? And hence the name. Let me read what I have on my slide. It's a pretty thick layer, 8 to 10 cell layers thick. It has a prickly appearance. The cells have this polyhedral shape. They're not squamous yet. They're still kind of roundish. The reason why the cells look like pincushions is they're producing a pre-keratin-like protein called tonofibrils. And that kind of gives the cells in this layer this kind of prickly appearance, hence the name stratum spinosum. Cells produce tonofibrils, which is basically a pre-keratin gives the cells a prickly appearance. Prickly. Cells are all connected by desmosomes. They're all linked together. It looks like a very spiny layer. And well, I kind of super zoomed in on our picture that we've been looking at. It's the same picture. I'm just kind of zooming in on different spots to show you the histology, how it compares to the illustrations in your book. But pretty much, more on the bottom is the spinosum. And as you get more towards the top, we're getting in the granulosome. The darker colored cells are stratum granulosum, but here it's hard to tell. They're supposed to have that prickly appearance. The cells aren't dead yet. Okay. But as you get pushed to the upper layer above this, they start to die. And that's stratum granulosum. As you get pushed to the top, they're squished and become squamous. It's like the cells are being squished to death. So it's this layer here, stratum granulosum. The cells are characterized by dark staining granules. So what's happening is they're producing a keratin and keratinohyaline that causes dehydration cross-linking that leads to the cell death. Cells above this layer die, basically. So because you're being pushed to the top, you're further and further away from the capillaries, the blood supply, so you just die. So I'll write that the keratin causes cross-linking in the cells. Proteins cross-linking in cells always leads to cell death. You just always see that. So leading to cell death is what's going to happen there. So cells above this layer will be dead. Okay. Too far from the capillaries. Anyways, let's see here. Oh, this is a good picture. So this is showing you spinosum granulosum lucidum. And even with the lights on, it's easy to see what's what. I put my bracket lines. It's kind of arbitrary where it begins and ends, but for sure when you're up here and the cells are the darkest, that's granulosum. Then above that layer, they're dead. So this clear layer, it's lucid or clear, stratum lucidum, that's where you have some, it's a thin row of dead cells, basically. So I just put dead cells, and it's a transition layer between the granulosum below and the corneum above. There's the same picture again. So when you get to the top, the thickest part is the stratum corneum. On thick skin, it's the thickest layer. It's the 20 to 30 layers of cells, and the keratinocytes at this point, they're devoid of any organelles. They're just filled with keratin, and you have glycolipids that link the cells together to create water resistance. So I'm writing keratinocytes are filled, obviously, with keratin, and glycolipids link them together. So cells change from stratum basale at the bottom to stratum corneum at the top. It's accomplished by a specialized form of apoptosis. Apoptosis is programmed cell death. It's controlled cellular suicide. Nucleus organelles break down. Just reading from my slide. The plasma membrane thickens. This allows the cells to slough off, as in dandruff and dandruff. And you shed anywhere, to put a number to it, 50,000 cells every minute. And that's called desquamation. I learned that term before. So there is thick and thin skin. The difference would be stratum corneum. The stratum corneum, let's do it this way. Let me erase this. Thin skin versus thick. I'll just say thin corneum, thick corneum, and thick skin. That's pretty much the biggest difference. And the picture shows that. But also, you usually don't see a lucidum in thin skin. I don't know, maybe it's there, you just can't see it, but usually lucidum is absent in thin skin. So those are differences between thick and thin skin. In terms of the organization of the dermis, we did talk about it already. It's the epidermal ridges and the dermal papillae. Actually, there's a name for that dermal organization. Just drawing the dermis. Remember the top part, or the dermal papillae, and remember I said those were a loose CT. But everywhere below it, see from here down on down, was our dense irregular CT. And the name for it is the reticular layer of the dermis. So the organization, dermal papilla up top, reticular layer down below. That's pretty much it for the dermal organization. The epidermal ridges at least on your fingertips, that's pretty much enhancing your grip and is responsible for your fingerprints. Okay, here's a picture of the papillary layer and the reticular layer on our slide. And if I turn off the lights, you can kind of see that the reticular layer, the bundles of collagen are a little thicker. But when you get to the papillary layer, you can see more light shine through the collagen bundles. In terms of other topics, burns and cleavage lines, now that you know the layers, epidermis, dermis, hypodermis, burns can be categorized based on the layers that have been damaged. I mean, people kind of know that in everyday language that third degree is the worst, right? It's because you've burned all the way down to the hypodermis. So I'll just put this for first degree, you damage the epidermis. Second degree burn, you damage the epidermis, but also the dermis below. Third degree is all three layers. The epidermis, plus the dermis, plus the hypodermis. All the way down to the muscle, basically. Okay. So that's what this slide says, and it has some other descriptions of it. But basically, I just want you to know third degree burns all the way down to the hypodermis. And tissue damage caused by heat, electricity, radiation, chemicals, it kills cells. The immediate threat is dehydration because you've lost that waterproofing of the epidermis. So immediate threat to severe burns. Dehydration. I used to have a student that worked in the burn unit at Shriners Hospital, and she used to tell me, like, this is the only thing they worry about when they get them in on the chopper is how dehydrated they are. And so you bring in a patient, an easy way to estimate the severity of the burn and how much is called the rules of 9. As a percentage, what you see here is the two upper extremities. If you have lots of burning on both upper extremities, that's about 9% of the body surface. The trunk is about 18%, and 4.5 for the head, 9 and 9 for each lower extremity. Rules of 9. So that allows the clinician to kind of like say, okay, well, this much is burned. This is how much fluids we're going to need to prevent dehydration and shock. And burns in kids is very dangerous because their skin is still very thin and hasn't fully thickened yet, as in an adult. And noodle burns pose the most serious risk. Now, a lot of people may think it's the hot fluid, but really it's not really that. It's more the noodles, because the noodles, they pack in the heat. They hold on to the heat very well. And if you spill the noodles on you, it stays on the skin longer. The liquid just kind of bounces off the skin. So noodle burns are, and they make it so you're going to burn yourself, right? I mean, how do they shape that cup? They shape it like that, right? So it just tips over easy. Okay, I wish they made them like Yoplait, the yogurt, right? It comes like that. That would be better. Or it'd be better, I serve my kids noodles on a flat plate. And I don't want them to burn themselves. Well, anyway, noodles can be a cause of burns, and I have some stats on that there, but you can take a look at that if you want. Basically, you get the idea. It's
So if you burn yourself all the way down to the muscle,
yeah,
you and essentially you've destroyed nerve damage.
That's right.
There's no way to get it back, right?
Nerves can regenerate. the thing about the nerves that we see in the skin is they're nerve endings, and the cell bodies are somewhere else. So there's the process of where the cell body can regenerate the nerve ending.
Okay.
Okay, so it's all about the cell body. As long as those aren't damaged, you're fine. Think about like spinal cord injury patients. That's where the nerves are. The nerves, the body cell. Okay, so cleavage lines are invisible, but I guess they're important for surgeons because if you follow the cleavage lines, there's less scar tissue that'll form. Healing will be better. So I want to note, have you know, just cleavage lines shown here. Invisible, but for surgeons, it's important. If you make your incisions along the cleavage line, for better healing. I did mention the different types of sweat glands earlier. This is a good table that helps summarizes them. I don't really have anything to add to this. Just remember sebaceous gland, the apocrine, the eccrine sweat glands. I also call them merocrine because the form of secretion is different. So maybe we could note that. I think that might be worth right here. Eccrine, apocrine, sebaceous. These two are sweat glands. Sebaceous gland secretes sebum. Remember, the apocrine one, I said that one that causes the BO because of the fat and protein content of the sweat, uses the hair follicle. But the eccrine gland, its mechanism of secretion is exocytosis. The apocrine sweat gland, also exocytosis. The sebaceous gland uses the mechanism of holocrine secretion, where the entire apical row of cells burst to release the sebum onto the hair follicle. Okay, that's a nice figure. Let's move on. So appendages of the skin include hair and nails. We'll take a look at those. The anatomy of a nail. It's not too complicated. I'll just kind of like list the terms that you may not have heard of before. When you look at your nails, they're pinkish underneath. And there's a little pale crescent at the base of the nail called the lunule. Pale crescent. There's less blood vessels there, so it's more pale than the rest of the nail. Okay. That's the term. Pale crescent lunule, looks like a half moon. But your nail lies in what's called the nail bed. The cuticle of your nail is also called the eponychium, and then underneath the free edge of your nail is the hyponychium. So eponychium, also known as cuticles. Hyponychium. This is pretty much where dirt accumulates on your nails. Kind of a weird word, but the word root right there means nail. I forget if it's Latin or Greek, but eponychium, hyponychium. Know the lateral nail fold. On the sides of your nail. Okay, that's good enough. That picture is pretty self-explanatory. The hair follicle, I showed you earlier. Hair follicles are infolding of epidermis, and the epithelial mass surrounding the hair bulb is the hair root. So if you look at the picture there, here's the epidermis up top. It just kind of folds and kind of surrounds the hair follicle all the way down deep into the dermis. The hair root, you would have living cells. But the hair you see in the hair shaft is just dead protein. There's no living cells. The hair root has living cells. So I'll just write hair root has living cells. There's a lot of pictures in your book that teach you the structure of the hair follicle. Pictures like this, and I kind of zoomed in on one. And I'm pointing to the different regions there. I kind of color-coded to make the font match the arrow. I'm starting at the bottom, called the hair papilla. This is the bottom of the hair root. It's connective tissue, capillary nerves, so it's the blood supply of the nerves in there. That's why when you pull a hair out, it hurts, because there's a nerve ending inside the hair papilla. So it has blood vessels, nerve ending. Now, surrounding the hair papilla is the hair bulb, and that's where you actually have the dividing cells that make hair grow. Okay, it forms a matrix, and the daughter cells undergo keratinization. Hair bulb. This is where you have the youngest dividing cells that become keratinized. Youngest dividing cells that become keratinized. So as hair grows, the young cells in the middle are getting pushed to the outer part. So right in the middle is called the medulla. The cells are lightly keratinized, and as they get pushed to the outer layers, they become more keratinized. So where I have that purple arrow pointing right to the middle is called the medulla. Where the cells have soft keratin. In the layers surrounding the medulla, that's color-coded here, I'm pointing to it with like green arrows. You have the cortex and the cuticle where the cells have harder keratin. Cortex/cuticle, hard keratin. All right, so outside the cuticle, we have what's called the internal root sheath and the external root sheath. Let's see here. Okay, let me erase what I have here. Start at the top. The whole thing is called epithelial root sheath. It's subdivided into internal and external. So I know it's hard to see in the figure. Let me point it out. It's pointing to this one right there and right there, these two, the internal and the external. And they're both called epithelial root sheath, these terms right there. The internal root sheath is lightly keratinized, and this layer even disintegrates to make room for the sebum. Okay, so lightly keratinized cells disintegrate to make room for sebum. So that's not shown on the picture, but it was shown on the micro model. See if I can zoom in a little more. So look for the internal root sheath on this model. Sorry. It's right here. There's the internal root sheath, and then there's the sebum, and then it's gone, right? Because it disintegrates so the sebum can kind of get on the hair follicle. All right. Back to this one. The external root sheath participates in hair formation not at all. It just does. It's a sheath. It has nothing to do with the hair formation. External root sheath. I just kind of think of the external root sheath as just a sheath. It's literally an infolding of the epidermis that's surrounding the hair follicle. Infolding of epidermis does not participate in hair formation at all. That's what a sheath is. It's just a sheath. Now, because it's basically epidermis, and all epithelial tissues have a basement membrane, what they call glassy membrane on the figure is a specialized basement membrane for the hair follicle. So on the picture, the glassy membrane is the bluish connective tissue surrounding it. Here's the term right there on the figure. If you're looking for it, and it's pointing to just this bluish connective tissue surrounding. So just know that glassy membrane is basement membrane. They also have a cross-section of the hair, but it's the same information. I won't reteach it. The only thing I would add that I haven't mentioned yet is the connective tissue sheet at the very bottom of this figure. The layers all match to what I said before. And pretty much the connective tissue sheet is derived from the dermis, and it surrounds the glassy membrane. All right. Connective tissue sheet. Surrounds glassy membrane. And derived from dermis. So I'm sure that's more than you ever want to know about a hair. So pull out your little hair and say, gosh darn it, there's so much. There's a lot of histology there. But I want to take a break. When we come back, we'll take our lab quiz, so be ready for that. I'm sure some of you would like to review. I'll give you a little more time on this break. Why don't we come back at 9:15. As soon as you come in, boom, quiz.
Is it multiple choice or?
No, fill in the blank. Which means you have to spell it correctly. See you at 9:15.